Hello guys, welcome to my YouTube channel. So today we are going to discuss one of the most important topics in plant biotechnology, which is Ti plasmid. So as you know, the Ti plasmid is found in a particular bacterium which is called as Agrobacterium tumefaciens, and it causes a specific disease in plant bodies which is called as a crown gall disease. Okay, and the total plasmid is of 200 kb. So this is the Ti plasmid which is specifically involved in the crown gall disease. Okay, and this is found in this bacterium. So if on the left hand side you can find a detailed diagram of a Ti plasmid. So as you will see, a portion of the Ti plasmid here you will find which is called as a tDNA. So from here until here, so this portion is the tDNA, and there are other things apart from the tDNA which is involved in the Ti plasmid. So just to resolve your confusion, Ti plasmid is actually made up of tDNA plus other genes. Okay, now this tDNA is actually the portion which is actually transformed and integrated within the genome of the host plant. Okay, so this is actually the portion which is transformed and integrated within the genome of the host plant and is actually involved in the development of the tumorous phenotype. So if we have just a quick description of the different genes present in Ti plasmid, you will find there are certain left border sequences and right border sequences which mark the boundary of the tDNA in the Ti plasmid, and there are other genes like auxin synthesizing genes, auxin coding gene, cytokinin coding gene. Now why auxin and cytokinin present? So these two genes encode plant hormones, so they help in the development of the tumor on the plant body. Okay, so the plant needs to synthesize certain plant hormones for the development of the tumor. So that hormones are provided by these two genes which code for auxin and cytokinin, and they come itself from the tDNA when a tDNA, or more specifically when that bacterium infects the plants. Okay, and apart from that you will find an opine gene. Okay, so opine is basically combined term for two catabolism gene products which are called, on one is octopine. Okay, on one is octopine and the other is nopalin. One is octopine and the other is nopalin. Okay, so these two terms are combinedly called as opine synthesizing gene. So now you have to remember that this genes opine either produces the product octopine or the nopalin. So in some strains of Ti plasmid you have octopine producing genes, in other strains you will have nopalin producing genes, but there has not been any report where you will find both. So either you will have octopine or you will have nopalin. Okay, now you may ask why this opine genes is important. So these are actually derivatives of amino acids and sugar phosphates which provide the nutrients for the survival of the bacteria. Okay, so which provide the nutrients for the survival of the bacteria. So these opine catabolism genes are the bacterial sources of nutrients, and they are derivatives of amino acids or sugar phosphates. Okay, so this point is very important. And these are opine catabolism genes. So first, from the opine synthesizing genes you have the opines produce either octopine and nopalin, and then the catabolism genes, what do? They break down the opines and nopalin so that the bacteria can utilize them. Another thing, you have one virulence region which produces some virulence products. Okay, virulence, which codes for some virulence protein, virulence genes, the VIR D, VIR B, etc. So these virulence genes are actually involved in the transfer of the tDNA. So what do? They involve the transfer of the tDNA, or they facilitate the transfer of the tDNA from the bacteria to the plant cell. Okay, so And another thing, of course, you have a separate origin of replication. Now this is very interesting. When in a plasmid, or particularly this kind of plasmid where the bacteria is having everything, okay, so it is having its own source of nutrients, genes, it is having the genes for the development of the tumor also, okay, so it is having everything, it is having its own virulence genes, it is having its own origin of replication. So this plasmid is very interesting and is of particular importance in biotechnology, more specifically plant biotechnology. Why? You can manipulate this plasmid and introduce your gene of interest into the plant body. Why? Because you have your own virulence region which would code for virulence proteins, which will facilitate the smooth transfer of the recombinant plasmid into the plant cell, and you have its own origin of replication. So if you introduce your gene of interest somewhere over here, suppose somewhere in the tDNA, okay, because actually it is actually the tDNA which is getting transferred. So if we introduce your gene of interest somewhere within the tDNA, with the tDNA it will automatically get transferred from the bacteria into the plant cell, because it is having its own origin of replication, so it will also replicate. Okay, so this is particularly interesting. So this way you can generate recombinant plant cells. Okay, by introducing recombinant DNA you can generate your own desired recombinant plant cells and help in the expression of your recombinant products within the plant body. Okay, so we will come to some particular characteristics of Ti plasmid. One, it can have one or more tDNA regions. This is very important. So whenever you get question, this is not something that it has to have only one tDNA region. It can have multiple tDNA regions. It should contain one VIR region. It should contain its own origin of replication, and it should contain sometimes a region enabling conjugative transfer. And apart from that, it should contain catabolism of opine genes. So opine catabolism genes, because these opines are actually the sources of bacterial nutrition, and when during the infection and during the development of the tumor, these opines are the only source on which the bacteria will survive. So now we come to the detailed description of tDNA. So tDNA, if you remember here, see here, tDNA is determined by the presence of right and left border sequences. So these are the right and left border sequences. Okay, and these sequences are 24 base pair imperfect repeats. So the repeats are not perfect. Okay, so you will not find any particular pattern. So as I have already told you that there are opine synthesizing genes, so it will code either for octopine or nopalin, but not both. Okay, so those Ti plasmids which code for octopine contains sometimes an overdrive or enhancer sequence. Okay, so in addition to the right border sequences, octopine strain Ti plasmid contains an overdrive or enhancer sequences, which is sometimes required for optimal tDNA transfer. So it helps in the smooth transfer of the tDNA from the bacteria into the plant cells. And those Ti plasmids which contains nopalin, okay, they have only one tDNA of approximately 20 kb length, and octopine strains Ti plasmids have two tDNAs which are termed as TL and TR, okay, and they are approximately of 14 and 7 kb respectively. However, only the TL region is oncogenic. Okay, so this is very important. This point is very important. So those Ti plasmids which contains nopalin, which codes for nopalin, have one tDNA, and those Ti plasmids which codes for octopine, they contains two tDNA. However, only one tDNA, and more specifically the TL tDNA region only is oncogenic. The other one, TR region, is not oncogenic and it codes for opine biosynthesis. So on the right hand side, you will find the structures of the two opines, octopine and nopalin. So as I have already mentioned, these are actually derivatives of amino acids or sugar phosphate. So this is derivative of pyruvate, arginine and pyruvate. Okay, and this one is derivative of arginine and alpha ketoglutarate, right? Alpha ketoglutarate. So another point that you should remember is the tDNA region of nopalin Ti plasmid contains 13 ORFs, while the TL-tDNA region of octopine plasmids contains only 8 ORFs. So this is also very important. Now, although there are certain points of dissimilarity between the nopalin Ti plasmids and the octopine Ti plasmids, in some portion they show extensive similarity, which is called as the core region, and this core region is actually the region which codes for certain proteins involved in hormone biosynthesis, more specifically the oncogenes, opine synthesis, and for determining the tumor size. So in the core region, okay, there may be different dissimilarities, like one nopalin strain Ti plasmids, the length is 20 kb, while octopine strains it is divided into two portions, 14 kb and 7 kb. Nopalin strains contain 13 ORFs, octopine strains contains 8 ORFs. So there may be many dissimilarities, but in a certain region there is extensive similarity, which is called as a core region, and it produces some proteins and it determines the tumor phenotype. Now we will come to specifically the oncogenes, and we will know why they are referred to as oncogenes. They are referred to as oncogenes because they are responsible and determine for the development of tumor phenotype. Okay, how the tumor will occur, what size, everything. Okay, so that's why they are called as oncogenes, because they help in the, or they facilitate in the development of the tumor. So there are two oncogenes, oksA and oksb, which encode proteins involved in the synthesis of oxines, indole acetic acid. So here I have shown you the TL-tDNA. So if you see here, this is the left border sequence of a tDNA, this is the right border sequence of the tDNA, and this is the ox gene. Okay, and this here you will find either oxa or oxb, okay, whichever you can find, but these genes are involved in the production of oxine or indole acetic acid. Okay, so oxa encodes tryptone monooxygenase and oxb encodes for indole acetamide dehydrolase. So this is very important. If you have read about the biosynthesis of indole acetic acid, oxine, you will remember that these two enzymes, tryptophan monooxygenase and indole acetamide dehydrolase, these two enzymes are central enzymes for the biosynthesis of oxine. So you need these two genes, oxa and oxb, within the tDNA for the synthesis of indole acetic acid. There is another gene which is called as a CYT, site, or TMR sometimes, or IPT, sometimes people refer to by different names, which encodes for isopentenyl transferase, and it is involved in cytokinin production. So if you remember, I have told you that within the tDNA there are genes involved in the synthesis of oxygen and cytokinin. So they are talking about this thing. Here you will find this site gene which will code for cytokinin. So these genes are primarily referred to as oncogenes because they are determinants of the tumor phenotype. However, the TR region of the octopine tDNA contains no such genes, no such oncogenes that we have all discussed, and that's why it lacks oncogenicity. So if you remember, only the TL region of octopine strain Ti plasmid is oncogenic. Now you can understand why only the TAL region is oncogenic, because it contains all these genes, oxa, oxb, site. Okay, and that's why only the TL region is oncogenic, while TR region does not contain any of these, and so it is non-oncogenic. Okay, so we will discuss some other genes. So genes involved in the production of opines are also present here. Okay, so might be present here, and not only these two opines, sometimes it has been reported that there are other opines instead of octopine and opalin, which are manopine, agropine and fructopine. They can also be present. So there is a third gene which we have not discussed. This is the TML gene. So this is the TML gene portion, and this is responsible for determining the tumor size. So whether it will be small, large, what phenotype it will occur. So this is determined by the TML gene. Okay, now we come to the VIR regions. So if you remember, so here this one, so VIR regions means the virulence region. So Ti plasmid contains one virulence region also. Okay, and this virulence region helps in the transfer of the tDNA from the bacteria to the plant cell. Now we will discuss this. So the genes responsible for the transfer of tDNA into the host plant is the virulence region, and it is approximately 40 kb in length. So there are roughly at least 9 VIR gene operons have been reported. Okay, and the most important 9 ones are this one: VIR A, VIR G, VIR B1, B2, VIR C1, VIR D1, VIR D2, VIR D4, VIR E1, VIR E2, VIR F, and sometimes VIR J. Okay, so more specifically, these VIR operons help in the passage of the transfer of the tDNA from the bacterium into the plant cell. So this is a very long process, and if you like have watched my video on like how the tDNA is infected and transferred within the plant cell from the bacterium, then you will get to know the function of each of these 9 VIR operons.